Good morning everybody and welcome to my channel.
In my previous video, I have completed the anatomy of cornea up to the stroma. Okay, so today we will start from the Duas layer. Okay, so let's start. So the Duas layer is the layer of the cornea of recent origin. So it is discovered by Dr. Harvinder Dua in the year 2013. The thickness of the Duas layer is 15 millimeter, and this Duas layer is made up of the collagen protein, and mainly the type 1 collagen is found in the Duas layer, although the type 4 and type 6 collagen also found in the Duas layer. But this Duas layer is purely a cellular layer because no keratocytes cells is found in this Duas layer. Okay, and this Duas layer is very strong layer, and air can pass through the epithelium, Bowman's membrane and stroma, but air cannot pass through this Duas layer. So this layer is impervious, very much resistant to the air. Okay, so next, the Descemet's membrane. The Descemet's membrane is the elastic membrane, and it is a homogeneous membrane also. So the term homogeneous means that the density of the constituents of the Descemet's membrane is similar or is equal throughout the membrane. Okay, and this Descemet's membrane made up of collagen and glycoprotein. Okay, and the thickness of this Descemet's membrane varies with the age. It means that the thickness of the Descemet's membrane is 0.3 micrometer at the early life, and its thickness is 10 to 12 micrometer at the adult age. Okay, and this Descemet's membrane is very strong and very much resistant to the chemical agents, trauma, infections and pathological process. So even when this whole of the stroma is sloughed off, means when the whole of the stroma is destroyed, the Descemet's membrane can maintain the integrity of the eyeball for a longer time. And unlike Bowman's membrane, this Descemet's membrane can regenerate when it is destroyed. Okay, so after this Descemet's membrane, we will go to the last layer of the cornea, that is the corneal endothelium. So this corneal endothelium layer is made up of the hexagonal cells. Okay, and this corneal endothelium layer is viewed by the slit lamp biomicroscope with the help of specular reflection illumination technique. Okay, the cell density of this corneal endothelial layer at the time of birth is 6,000 cells per millimeter square. Okay, and when it is viewed through this slit lamp biomicroscope, this corneal endothelium looks like a mosaic. So this pattern is mosaic. So what is a mosaic pattern? It is a regular pattern by arranged together the small tiny cells or small tiny particles. Okay, so with the increasing in the age, the number of the hexagonal cells in the endothelium layer is decreasing. Okay, and we will study that this corneal endothelium has performed different important actions like corneal barrier functions, active pump mechanism, which is very important for maintaining the corneal hydration. So if the number of the cells has been reduced, the important functions should be affected, but in reality these functions is not affected. Why so? As because this corneal endothelium has a unique property, is that the defect which is produced by the dying cells is compensated by the enlargement in the diameter of the existing cells. Okay, so if you look at this example, suppose let's consider this is a 1 mm into 1 mm square, and at the time of birth here is the 9 cells. With the increasing age, the same area is covered with the 7 number of cells, so the number of cells is reduced. So let's suppose these 2 cells have been died. Okay, so this defect which is produced by these dying cells is compensated by this enlargement of these cells. So these cells is now getting enlarged into this, so mainly the diameter of these cells is getting enlarged. Okay, so the diameter of the endothelial cells is about 18 to 20 micron at the early age, and it increase up to 40 micron at the later age. Okay, the cell density of the endothelial cell with the increasing age is can decrease up to the 2000 to 3000 cells per millimeter square. Okay, so the functions of the endothelial cells is the barrier functions. So these endothelial cells are attached with the previous Descemet's membrane and the other endothelial cells by the tight intercellular junctions, which are called as the desmosomes and also the zonula occludens. Okay, this tight intercellular junction results in the barrier function of the cornea, and also these endothelial cells are plays an important role in the active pump mechanism of the cornea. Okay, so look at this picture. This is this corneal stroma, this is the Descemet's membrane, and this is the endothelium. Here these are the ion based pump mechanism which are actually pumped out the excessive water from the cornea. Okay, so for this ion based pump mechanism, this corneal endothelium layer needs energy, and this energy is provided by the metabolic activities of these corneal endothelial cells. So the presence of abundant mitochondria, free ribosomes, smooth surface endoplasmic reticulum, Golgi bodies or Golgi complexes in the cytoplasm of this endothelial cell is the proof that the metabolic activity has occurred in these cells. Okay, and after the rods and cone cells, the endothelial cells contains the highest number of mitochondria. Okay, and one important thing about this endothelial cell layer is that the increase in the diameter of the existing endothelial cells is called as the polymegathism. So you can look at this picture. So in this is the normal corneal endothelium. Okay, and in this picture, so these cells are have been dyed. These cells have been dyed. Okay, so these cells are getting enlarged in diameter. So this phenomenon is the polymegathism. Okay, this is all about the detailed anatomy of the cornea. Let's summarize all these things at once. Okay, we will start the anatomy of cornea. We will understand that what is cornea and the three coats of the eyeball, why this cornea is also called as the watch-glass like structure, the basic properties and anatomical dimensions of the cornea, the different layers of the cornea and their terminologies, the detailed explanation about the epithelium and its different cell layers, the Bowman's membrane, the detailed explanation of the stroma, what are the constituents of the stroma, and about this peculiar arrangement of the corneal lamellae or stromal lamellae. Then we will completed the Duas layer, the Descemet's membrane, and this corneal endothelium today. Okay, and we have already discussed what is polymegathism, what is the active pump mechanism, and what is the unique properties of the endothelial cell layer. Okay, thank you for attending my lecture. Thank you very much.